So basically, we are studying biochemistry subject. In biochemistry, we started a topic called carbohydrates. In carbohydrates, we discussed about monosaccharides. Okay. So today we are going to talk about the further second related saccharides, that is disaccharides. Okay. So we have disaccharides. What does mean disaccharides, and what are the important things we have to understood about that? We will discuss in this class. Oligosaccharides, these we have single sugar cell or single carbon cell. Okay. In disaccharides, we have more than one, 2 to 10 carbons are present. Okay. Here, two to more than two. Okay. So this is the thing we have to understand. And they are crystalline. Okay. They are crystal like substance. Okay. And they are water soluble. If you use disaccharides, it can easily water soluble. Okay. And if you want to taste those disaccharides, it is sweetest in taste. Okay. So basically, there are two types, reducing and non-reducing. In the reducing, the example is given here, maltose, lactose. Okay. In the non-reducing, we have sucrose and terhalose. So we'll discuss each and everything. So here first, the given is maltose. So it have two alpha D glycose units. Okay. Glycosidic bond. So they are the chemical things, chemical related things you can see. Here we have the glycerol, myo, inositol, everything is here. A diagram. Okay. Okay. So maltose, in that we have two alpha D glucose. That's the reason this is different from lactose. This is different from sucrose. This is different from terhalose. Okay. The difference is the composition. Okay. So this the we have to think, remember. Next we have sucrose. Sucrose is also known as cane sugar. Mostly in the maltose and lactose, we have same type of uh, 2 alpha D glucose. Due to that reason, we attach in reducing thing, and the reducing is in the disaccharides. Okay, sucrose. Sucrose is also known as sugarcane or sugarcane or cane sugar. Okay, and it have, what does it have? Sucrose is made up of alpha D glucose. Here it is made up of alpha D2 glucose, alpha D glucose, and beta D fructose. In that we have the amount of the sweetness, and that is increased. Okay. So in the sucrose we have more carbohydrates. In the maltose we have less comparatively sucrose. Okay. And it is also as it is non-reducing sugar. It cannot form osazones. Sucrose is an important source of dietary carbohydrate. It is sweeter than most other common sugars. Okay, also we know that because of the presence of alpha D glucose and beta D glucose. That's the reason it is most sweeter than other uh, things like fructose. Sucrose is an employed as a sweetening agent in food industry. So most of the food industry utilize sucrose as a agent to give sweetening, whatever uh, product we use. That we have some common products like Horlicks or any other uh, thing that is that we eat. Okay, mostly in sweet also. Okay, in the intestinal enzyme, sucrase hydrolyzes sucrose to glucose and fructose, which are absorbed. So basically, whenever you take sucrose, what will happen? It will divide it. our body, in the intestine, we have sucrase is an enzyme which divide the sucrose into glucose and fructose. Okay, this is very important. Inversion of sucrose. So, so after that, the absorption is happened. Okay, if it doesn't divide, then sucrose will all remain and it will excrete, but it will not happen. But it is divided into glucose and fructose. Glucose is also absorbed and fructose also absorbed. It is an important thing we have to remember. And this sucrose is converted into sugar also, invert sugar. What we are eating sugar, okay, for making uh, tea, coffee, okay, or homemade sweets, everything. Okay, those are things will made from this sucrose only. What they do, they will hydrolyze, means whatever amount of hydrogen is there, it will excrete, it will decrease from that. Okay, so we they use sucrase enzyme, which is also present in our intestine. Okay, it will dilute acid, liberates one molecule each of each glucose and fructose. Okay, all this, this is the chemical processes they will do. Okay, to convert the cane sugar into simple sugar or invert sugar, whatever you will call. This is the process. Okay, next we have, after that, lactose. What is lactose? This is known as milk sugar, since it is disaccharide found in milk. Okay, so if you taste the milk without sugar, you can easily, without boiling. Okay, after boiling, the it is processed and everything is vaporized. Okay, if without boiling the milk, in olden days, most of the our father, grandfather, they used to drink, okay, without boiling the milk, because the presence of lactose in that, okay, and other um, vitamins and other protein substance which can easily available after boiling. So the it will vaporize, all the important elements present in the milk will vaporize. So best, but not nowadays. Nowadays we are getting injective or steroidal milk, or sometimes duplicate milk also are getting by, with the help of chemicals. They are producing milk which is smell like milk, which easily boil, which not break. Okay, okay. Sometimes most of the people will take. Okay, but here lactose, in the lactose is present in milk. Okay, without boiling we will get lactose. So without boiling, if you drink the milk, what will happen? You will get sweet taste. You can easily drink. After boiling only, the it will become very some sort of taste will change. Okay, because of evaporation of all these elements. So, so we have to understand that milk having lactose. In that, what is the composition? Beta D galactose and beta D glucose. So both will become attached and will make lactose. Okay, so this is their bonding and all the chemical things. Lactose of milk is the most important carbohydrates in the nutrition of young mammals. It is hydrolyzed in intestinal enzyme lactase. So all these enzymes are secreted by pancreas, lactase, okay, to glucose and galactose. So the lactose, uh, it is converted into carbohydrate, because it is very important for small children, okay, pediatric, okay, because they need milk as a energy source. Okay, who will convert into energy source? In the intestine we have one enzyme that is called lactase. Okay, amylase, lactase, all these enzymes are present in the small intestine, duodenum. Okay, and who will release? Pancreas will release. Okay, to digest. Digestion means to break down larger molecules into smaller molecules and made them to absorb by small intestine and utilized by our body. Okay, so what lactase will divide lactose. Lactase will divide lactose into glucose and galactose. Further, dividation also happens. Okay, so we talked about here lactose. Now, next, next is lactulose. This is an another element, another, but it is synthetic. It is combination of galactose and fructose. It is not digested nor absorbed in the intestine. Okay, because the food ever any person is making outside, it is not available in the natural ingredient. It is not a natural ingredient, natural substance or natural element. It is synthesized. It is made by two other sources, that is galactose and fructose. Thus, the reason it is not digested, not absorbed in the intestine. Lactulose is usefully for the treatment of hepatic encephalopathy. So, this is infection in the liver. Hepatic means liver. Encephalopathy means infection of liver. Okay. So, whenever the person is having encephalopathy of liver, they will treat, the modern medicine will treat with this lactulose. How this is happening? Okay. How they are treating? Yeah, disorder characterized by elevated plasma ammonium levels. So ammonia levels are increased. Okay, due to this ammonia level, uh, we have the liver is damaged or infected. So what will happen? Lactulose will release all this ammonia and decrease the ammonia, and it will not help to uh, absorb those ammonia in the bodies, because ammonia is very very dangerous. It will cause very serious injury, serious damage in the brain, and it will stop the entry of oxygen and RBC to the brain, and it also damage HB or iron. Okay, all this will happen. So that's the reason they will give lactulose, which will reduce the effect of ammonia and it will release the ammonia from the body. Second, primarily, and it will could not affect any brain particles, brain cells. Okay, so this is all about disaccharides. So disaccharides have two things, that is reducing, non-reducing. In the reducing, we have maltose, lactose. In the non-reducing, we have sucrose, trehalose. Okay, so we'll continue. Next, we have that polysaccharides. So kindly give please 1 minute. We will resume. Yes, sir. What are you asking?
Sir, that's why this lactogen given to children.
For what purpose? Purpose?
This lactogen food.
Yeah,
This
yes, yes. Because it is helpful for growth, for production of energy, because it's also present in the milk. Okay, lactose.
okay, sir. Okay, okay.
Okay. So we completed disaccharides. Now we have polysaccharides. Poly means more than 10. 10 carbons are there. Okay. So there are two types of polysaccharides. One is homopolysaccharides and another one is heteropolysaccharides. Homopolysaccharides means is only a single type of monosaccharides. They are namely based on the nature of the monosaccharides. Thus, glucans are polymers of the glucose base, fructans are polymers of fructose. Means monopolysaccharides means in that we have different hydrogen molecules, sorry, carbon molecules, but they are belongs to same family. Okay, hetero means They are mixture of different carbon families. Okay. Monopolysaccharide means they belongs to same family. Hetero polysaccharide, they belongs to different types of families, but they have same more than 10 amount of carbons. So here we have examples. Homopolysaccharides. In the homopolysaccharides, we have starch. Okay. And next we have dextrins, dextrans, inulin, glycogen, okay, cellulose. All these are derivatives of homopolysaccharides. We will discuss one by one. In the starch is the carbohydrate reserve of plants, which is the most important dietary source for higher animals, including man. High content of starch is found in cereals, roots, tubers, vegetables, etc. Starch is homopolymer composed of D-glucose in glucose units held by alpha glycosidic bonds. It is known as glucose or glucan. So basically, starch is a high amount of rich carbohydrates which is present in the plants, related to the plants like cereals, beans, the roots, vegetables or leaves. Okay, so whatever we are consuming, that we will named as starch of that carbohydrates. Okay, because of presence of high amount of carbohydrates in the plants, that is named as starch, because that is monosaccharides, and the first one is homopolysaccharides. In that we will able to get one type of same type of molecules in multiple belong to the same plant, same family, but their levels are increased. Okay. So starch are derivative from plants, animals, sorry, plant related things, cereal, roots, vegetables, everything. Here we will get a few thousand glucose units looks like a branch tree. Okay. So here, so that is starch. Starch are hydrolyzed by amylase. So we have amylase, lipase, lactase. These are the enzymes which released by the pancreas, and their hydrolyzation means broken. Okay, starch has broken in the small intestine. Okay, this is the thing we have to. So, to liberate dextrins and finally maltose and glucose units. So, they convert into dextrins, maltose, and glucose. Okay, so here we have dextrins. Dextrins are the breakdown product of, so, and starch who will do amylase or dilate acids. Okay. Finally, to the maltose and glucose. The various intermediate are soluble starch. Okay. So these are the different types of starch. So basically, dextrin is the derivative from the starch. So we have to remember that. Okay. Thing. Next, we have dextrans. Dextrans are polymers of glucose produced by microorganisms. They are used as plasma volume expandation transfusion chromatography. Okay, say what dextrans has that we have glucose. They are the glucose polymers. Okay, but here what we have to understand, that is uh, monosaccharides, not monosaccharides, polysaccharides. Okay, so we are understanding the examples of polysaccharides, that is dextrans. Okay, next we have inulin. Inulin is a polymer of fructose, so it is a combination of fructose, like if where it is present, isn't present in dhalia, garlic, onion. Okay, its weight is very low. It is soluble in water. Inulin is not utilized by the body. It is used for ascending kidney function through measurement of GFR, glomerular filtration rate. So how your kidney functioning is happening, so inulin will decide. So it is the inulin that the agent will means it will uh, which will help to know the amount, the actual function of kidney. Okay, it is a polysaccharides. In that polysaccharide, we have homopolysaccharides. Next, we glycogen. Glycogen is also the conversion of glucose, and large molecules of glucose are attached and stored. So in that also we have more than 10 glucose molecules. So that's the reason we are calling polysaccharides. In that same type of molecules are there like glucose. There is no lactose, fructose. There is no thing that we have glycogen in the glucose only. Okay. So it is also stored in the liver, muscles, brain. Okay. And the glycogen is also present in the plasma. Okay. Of the plants or plants, covering of the plants. So okay, this is the thing we have to remember. Next is cellulose. What is cellulose? Cellulose means there is the most abundant organic substance in plant kingdom against starch, glucose, cellulose, everything are from plant because we eat the plants derivatives, their roots, their stem, their leaves, sometimes their flower, sometimes their fruit. Okay, cellulose is totally absent in animal body. Okay, it is only plant. Cellulose is composed of beta D glucose units, so it is made up of beta D and beta D glucose units. Okay. Cellulite cannot be digested by the mammals. Mammals cannot be utilized this, including man, due to the lack of enzyme that cleaves beta glycosides bonds. Certain ruminants and herbivorous animals contain macrophages, microorganisms in the gut, which produce enzymes that can cleave beta glucosides. That bonds hydrolysis of cellular yields a disaccharide cellulobiosis bios followed by beta D glucose. So this is about cellulose. Cellulose, though not digest, has a great importance of human nutrition. It is a major constitution of fiber. So what is the whatever product having cellulose, it means the great having the great amount of fiber. So fiber it is cannot be absorbed in the body. It is not digestible. It is a helpful for excretion of waste material. Okay. What it will do it that it is not digest non-digestible carbohydrates. The function of dietary fiber include cholesterol from the intestine beside increasing the bulk of feces. Okay. So this is the cellular. Next we have chitin. It's composed of these things. It is structural polysaccharides. Okay, nothing about that. Chitin. Okay, so this is all about polysaccharides. Homo polysaccharides, and we have mono disaccharides, monosaccharides. All these are the different types, three types. So you will discuss it, or else, or we will go forward from tomorrow. Next, we'll discuss hetero polysaccharides from tomorrow. If any doubts, kindly ask.